Welcome to another Everything ALS Expert Talk Series. I want you to remember that you're a very important member of our team, each of you, because it's together that we can gain the wisdom to understand and defeat ALS. So thank you so much for being here. And with that, our founder and CEO, Indu Navar. Welcome, Indu.
Thank you, McFinn. And I have pleasure today of inviting and honored to have Dr. Lyle Ostrow to come in and speak to our community today. And Lyle is MD, PhD, is a neurologist and ALS researcher at Lewis Katz School of Medicine at Temple University. He's also a member of Scientific Advisory Board for Everything ALS and the Board of Directors of ALS Hope Foundation. He's a director of the Temple University ALS Postmortem Core, which is funded by the collaboration with CDC National ALS Registry. He's also director of the Temple Nerve and Muscle Biopsy Service and Neuromuscular Pathology Lab. Dr. Ostrow is the chair of programmatic panel for the DoD ALS Research Program, presently the largest dedicated annual funder of ALS therapeutic discovery and validation. He's helped lead the ALS-RP efforts to develop refined funding mechanisms to help novel treatments advance from the lab to clinical trials, emphasize biomarkers and clinical outcome development, and encourage open data and resource sharing. With that, I would love to welcome Dr. Lyle Ostrow on Blowing Up the Pipeline, Stories from the Front Line of ALS Revolution. Thank you, Lyle.
So I'm so grateful for the chance to talk to you guys. And I'm not going to talk about things that, my research. This is not about me, but I am going to talk a little bit personal, and I'll explain why in a moment about kind of the things I've been thinking about lately about what's going on in ALS and really what a privilege it is to be part of this community. So Indy mentioned, so I'm a neurologist, I'm a researcher. I think I know probably the majority of the people on here, and I'm grateful for that. I have research support. I have a grant looking at a particular ALS therapy with collaborators from NIH. I'm funded by the CDC National ALS Registry and Biorepository for our Postmortem Core effort. Indu mentioned that I'm the chair of the programmatic panel for the Department of Defense ALS Research Program. That's a volunteer position. I am not, in any way employed by. I don't speak for them. I don't speak for the CDC, of course, either. But that's something I do as a volunteer. And certainly I'm on the Scientific Advisory Board for Everything ALS and also the Board of Directors for the ALS Hope Foundation. And again, those are both things that I do have the privilege of doing as a volunteer. And so this is going to be a little bit kind of maybe informal, and I want to definitely have a lot of time to talk at the end, although those who know me know that I tend to talk too much, so we'll see how this goes. What I want to tell you about is why I'm here, and by here, I mean the big here. Why am I part of this community, and what am I doing here? And what do I mean when I say blowing up the pipeline? What's that about? And why am I calling these stories from the ALS Revolution? What's the revolution? And I guess what I want to start with is the true why am I here? Why am I part of Everything ALS? How did I become part of this program? How did I get to be part of this community? And about 2 years ago, you guys invited me to give a talk, right? And this was the talk I gave, and I said, we need to do better. We need to find better ways to identify and validate ALS therapies, right? And at that time, I was at Johns Hopkins. I've since moved to Temple. That's actually a pretty old picture of me. I had to shave the beard for COVID. But, what did I talk about that day? And I started off talking about this guy, Jack Griffin. So Dr. Griffin was the chair of neurology at Hopkins. When I came as a medical student, I worked with him in his clinic, and when I was a resident, and when I joined the faculty, and he's really, a real personal mentor as far as how to be a neurologist and how to think about things. And he also was the editor of research in neurologic diseases, of Nature Clinical Practice. And he wrote this editorial about research. And And when I talked 2 years ago, I started off talking about it. And the reason was, I pointed out that this was written in 2006, but, you know, it could be written today. And what I mean by that is that we still are impatient and frustrated. And even though we've learned so much about the biology of this disease, that hasn't necessarily translated into therapies. And, you know, we need to do better. And when I talked then, I talked about these specific things that Dr. Griffin had suggested about finding ways to focus on speed and you know, requiring unfettered exchange of ideas and data, and developing new intellectual property approaches that do not delay progress. And, you know, when I was first invited back to talk now, I was kind of uncomfortable about it. And at first I sort of, to be totally honest, I think I kind of blew you guys off for a few weeks and didn't respond because I was wringing my hands. And I really wanted to think about, you know, what can I talk about? And the fact is, when I sat back and thought about what's changed in those 2 years, it's amazing. It's amazing how different our you know, ecosystem and you know, our environment is, and how we're actually doing these things. And you were talking about some of it when I came on the line, but you know, things are changing, and they're changing fast. And it didn't feel this way a few years ago. It felt like there was this sense of, what's the next trial that's going to fail? And you know, it really has changed. And so I wanted today kind of talk about why I think that is. And, you know, this hasn't changed, right? This we all know. This is the problem specifically with ALS, that we have all this information from the lab and all this data, and yet the trials have, you know, generally not been successful. And we think maybe this is because, you know, a given model, a given mouse, a given, you know, cell culture may only be significant for a certain subset of ALS patients. And so we need to figure out, are we using the right treatments in the right people at the right times? You know, how can we do a better job of validating what we find in the lab in you know, people living with ALS, to make sure that we're doing the right therapies? And you know, and how can we tell if a treatment is working, right? What can we measure? And this is an example I've used before, and I give these sorts of talks a lot, and I'll get to why in a moment, but you know, this is an ALS mouse, right? This is an SOD1 mouse, a mouse that has a genetic mutation that resembles one kind of ALS. And these are all littermates, and these are ones that are treated with a drug, and these are ones that got saline. And if I play the video, you know, you can very clearly see, I think, that in general, the guys who got this drug over here seem to be moving around more, right? And so that looks good. Maybe this is going to be an interesting therapy, but you also notice right away how variable things are. Some of these mice are brown, some of them are black, some are big, some are small. One guy over here seems to be moving maybe better than the others, and walking around some. And so how do we know whether this is a useful therapy? And you know, we've learned a lot about how to use these models and how many mice do we need, and you know, how to do rigorous research in the lab. But we still need to make sure that we are when we translate these to people, that we're doing it in the right people. And so, you know, back then and in several talks since then, you know, these are the things we all talk about, right? We all talk about these frequently in lots of different avenues. You know, how can we do better? What really is ALS? You know, how do we find therapeutic targets? Why are some things in trials and some things not? And, you know, who chooses? Who decides that this thing is in a trial and that this thing isn't? And how does that work? And why can't we, you know, develop drugs like they do in cancer or in HIV? And how can we, you know, de-risk investments so that pharma partners aren't spending tons of money on failed trials? These are the things that we continue to talk about, right? And we've made some significant progress, but these are still the topics. What's a biomarker? Why do we need them? You know, how can we use this interconnected world we live in to do a better job of these things? And, you know, I mentioned I give these kinds of talks frequently, and you know, I often get invited to do this because of my roles with the DoD ALS research program as a volunteer, and also running this postmortem tissue core that I work with a bunch of researchers all over the world who are developing therapies. And so, you know, I often get asked to talk about those things. And to be honest with you, I usually don't talk about what I'm been asked to talk about. I usually end up talking about how none of this would be possible if we weren't all working together, and how that really is changing, right? And so this was a talk I gave actually, you know, in October 2022. At that point, I'd left Hopkins and hadn't started at Temple yet. So I was myself in some transition. But again, you know, how are we all working together? Why does it matter? And I mentioned, I blowing up the pipeline. What do I mean? I don't like this word. And one thing I'd like to sort of, I hope, convince you of today is we got to stop using it, and I'll explain why. But, you know, this was a talk. So some of you I know on this line have been part of the NEOS Clinical Research Learning Institutes. And I sometimes talk about the research pipeline and development of therapies and those. This was something I did again, you know, last month for the ALS Association, talking about federal appropriations, because, you know, I have funding from NIH and CDC, and I work with the CDMRP, the ALS Research Program. So, you know, I wasn't talking on their behalf, but I can talk as perspective of why these things are important. And I always say, and I really want to stress this, because I'm going to say some things today that are my own personal opinions. You know, I don't speak for these programs, right? I'm not an employee of them. What I say is my own opinions. But I couldn't do what I do without their support, and I'll explain why it matters. So this is a pipeline, right? Pipelines are an eyesore. They don't interact with their environments. They're a tube. Once upon a time, somebody said that this is how we're going to describe, you know, a pathway to therapy, because the idea was that something's going on in a lab, and then, you know, it gets developed, and then eventually it gets handed off to the clinician, and the clinician does the trial. And there really wasn't much interaction, I don't think, with the environment, with people living with the disease, with organizations, whatever it might be. You know, it was a straight shot until one day it appeared at a clinical trial. Maybe people living with the disease were asked to, you know, contribute money or something like that to help fund research, but that was really it. And, you know, I think a lot about how that's not how we operate, right? We don't do this anymore. This isn't the way that we develop therapies, and it's important. And so I really, really don't like this word. This is the other thing that pipeline can mean. I like this even worse. This is a pipeline, you know, a surfing pipeline. That's a wave that's about to crash on that person and make them wipe out and go underwater. And so, you know, I'm not fond of the word. And by the way, this is if you do a Google search for pipeline. This is not what I want our therapy development landscape to look like, right? This isn't what I mean. And so I want your guys' help, but I think we have to find a new word to describe what we do. And it's not just about the fact how it interacts with the environment. I do think, you know, when I think about it, like when a pharma company talks about their pipeline, there is something maybe implied in that this is our, you know, super secret thing that we're doing, and just trust us, and we're going to come up with the drug on the other side and a trial to help you. And that really isn't the way we operate anymore. You know, this is a slide that I borrowed from somebody at the ALS research program at one point, talking about how, you know, this pathway to therapies in ALS. And, you know, you start with risks and epidemiology, and, you know, what are the different causes? And that's really the purview in a lot of ways of the CDC ALS registry. And then, you know, basic biology by NIH, by other organizations. The ALS research program of the DoD has been firmly planted here in this you know, immediate preclinical and translational clinical development, trying to get new therapies into responsible trials. More recently, actually, in some early stage trials. And then we do depend still on industry funds for the later stage trials. And that's something I hope is going to change in the future. And we're going to maybe talk about that some. But you know, the fact is, every aspect of this pipeline has been majorly affected in the past few years alone by this community. Whether it's, you know, changing the way that the National Registry does their annual meetings, or you know, the kinds of grants that they're awarding, or funding for research, or, you know, increasing the funding in Congress, and helping with improve the design of programs, helping to improve the design of trials. At every stage, you know, it is people living with the disease, and advocates, and ALS warriors, and foundations that are really making the change. You know, I've been part of the ALS research program since, I guess, 2012. I was super proud of this, right? So Sandy Morris, who's passed away, but I know many of you knew her, was an unbelievable advocate, and came up with the Morris principles, nothing about us without us. And this was an award that you know, that Morris Principles gave to the ALS Research Program in 2022. Since I last spoke with you guys, I think, you know, it says right here, the CDMRP ALS Research Program included people living with ALS on all scientific advisory bodies related to research. Since its inception, it's included people with lived experience as consumer reviewers of research grant awards. I know many people on this call have done this. The scores and input of consumer reviewers carry equal weight to that of the scientific reviewers, a seat and voice at the award ranking table. At every stage of the ALSRP, it involves people living with the disease in deciding what kinds of things we fund, how we should design the mechanisms, review of the grants, everything, and it matters. And, you know, so again, I no longer call these talks pipeline. I call them whatever, challenges and opportunities. I know a lot of people here have done the Clinical Research Learning Institutes. This was a recent one at the ALS Hope Foundation. I joined virtually because I was actually out of town, but, and this is, by the way, from the newsletter of the Hope Foundation, but I know there's people in these pictures who have already, just in the past month, impacted this pipeline and are helping to raise funds and figure out ways to help get you know, biological samples and the rigorous things that are needed to move research forward. And so this is like, you know, it's unbelievable how fast this is moving. If I don't check my email for a day, I have fear I'm going to miss out on something important and exciting. I joined the ALS research program here in 2012. This was the congressional funding for that program at that time. When I joined, we would get, you know, 85 proposals and we could fund 3, right? And no matter what we did and no matter how well this was designed, it didn't really feel that, you know, meaningful. Because of work of advocates and you know, helping to redesign the funding mechanisms and make them better mechanisms and you know, therefore have them show that they are effective, we've been able to increase the funding. These numbers here, these are the numbers of grants, right? So look at this, starting in FY20, it went from, you know, anywhere from 5 to 10 million up to 20 million. And we funded 24 grants, 24 new ALS research projects. And then each of the past 3 years, you know, 40, 41, 30, like, you know, this is over 100 new ALS therapy development research programs in just 3 years. And it's just remarkable how much that's changed since even the last time I spoke to you guys. And it's all because of the fact that this program is completely the direction, everything that shapes it, it's the entire community. That first year in FY20, when this budget was going up, was the first year we offered an award dedicated to helping to define subsets, you know, make new biomarkers, find ways to improve clinical trials by identifying which people might respond to which therapies, finding better ways to measure disease progression. These were those first set of that new award from that year. You know, they were things like adding on, you know, biomarker development onto ongoing trials. Figuring out, so this year, examining the FUS ASO trial, you know, to look at novel markers that can be engaged for therapy. I'm sure most people on this call know that, you know, the gene therapies for FUS are doing remarkable things right now. It's the first time in my life that, you know, Dr. Schneider and the team at Columbia, that I've ever seen somebody living with ALS truly, you know, get better with a genetic therapy. And it's because we know which therapy to give to those people, the gene that they have a mutation for. I wanted to just, you know, some of these are analyzing publicly available biorepository samples that would not exist. These resources wouldn't exist if people living with the disease hadn't contributed their blood and their spinal fluid and their tissues and their data and all the rest of that. And now this is making it so that we can leverage those resources and do research on them to develop therapies. You know, Dr. Fournier at Emory University, she got an award to build this new thing that's now the ROADS, which I know you guys are familiar with, which is a new, you know, scale that we can use to assess somebody's progression with ALS. By the way, after getting this award, I think it was a year or two later, Dr. Fournier is now on the programmatic panel as another volunteer, helping to shape, you know, how this program moves forward. So, you know, that was back then. This is now. So now we've got, you know, $40 million. This is, if you go on the website right now, this is, I copy pasted these from some new resources on the website. These are just some of the therapies that literally over the past 2 years have been moving forward from early, you know, support of this ALSRP. None of this would have happened without people living with the disease. And you know, we've been able to expand the mechanisms. We still are most known for funding new, innovative, novel ideas, but we also really focus on biomarkers and on early phase trials, so that when something gets to the Healy platform, you know, that it has the information in hand to make it a good trial, to know whether it's working, to have the right biomarkers. I wanted to talk about this right now, really stress it, because guess what? This call is open right now. If you know ALS researchers, if you are part of a foundation, anybody can apply for this. You don't have to be an academic lab. It can be industry. It can be nonprofit organizations. It can be anywhere around the globe, okay? These are the mechanisms now. This is how this program has matured. Right now, the deadline, June 18th for a letter of intent, July 10th for the full applications. The funding rate for these mechanisms is better than anything in science. The one thing that we really are doing a bad job of is publicizing these, and we're trying really hard to get the word out. And so I'm asking you, please, 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 if you know ALS researchers, if your ALS clinic has some cool idea about the way that they're handling, secretions or respiratory care, encourage them to apply. We still fund these idea awards to fund innovative new ideas, no preliminary data. Development awards, these are to get the what are called IND enabling studies done, toxicity studies, manufacturing studies, everything that you need for the FDA to let you do a trial. We fund pilot clinical trials now, right? And these trials, they must include biomarkers. They must include mechanism-specific biomarkers. Researchers have to have a way to figure out which people should get their drug and to monitor in the trial that the drug is having the effect that it's supposed to have on the biology. And then, of course, we also have awards to help develop those markers, and including digital technologies, which certainly EverythingALS, has been very focused on. So better ways to track people as their disease progresses, better ways to define subsets of patients. With digital markers, with things that we can do remotely, we can involve people in clinical trials who don't live near brick and mortar clinics, right? This is all about equity in trials too. We can monitor people and see how they're changing over time. We can get data much more frequently. We can monitor at-risk gene carriers who don't yet have ALS and see how they're changing and do trials on these people that otherwise wouldn't be possible. And so please, if you know people who have great ideas, tell them to apply right now, today. We also, as this program has evolved over the past 2 years, so those clinical trial mechanisms absolutely require community collaboration. There must be people living with a disease or, a steering committee, some community collaboration that's helping with the design and execution of every clinical trial that's funded by this program. We also require data sharing. It's a requirement. We don't just require a data sharing statement like they do for NIH. We actually score it, and it's part of the evaluative criteria for these awards. So, the other thing that I spend my time on is, and this is what's funded by the CDC, is running a postmortem tissue core. And, all of this work on figuring out how to translate from the lab to people living with a disease, figuring out how to monitor a clinical trial to find markers in somebody's blood or their muscle or whatever it might be, that are changing when you give a therapy. This requires patient-derived samples and depends on it. And this is where all of the research comes from too, right? And so, the idea is that you want to be able to validate what we find in a mouse or in cells in something that's relevant to people living with a disease. And, when I give those sorts of talks, this is often I talk about that what we do in the lab, where all these mechanisms come from, is that you have two things you're comparing, right? And sometimes, you have your disease and your control. Sometimes they're obviously different, like an apple and an orange. Sometimes they're not obviously different, like two different species of apple, two different, types of apple. But if you grind those apples up and make applesauce and you analyze them, maybe you're going to find that they've got different amounts of sugar or different ingredients. And, when we think about the brain is more like apple pie because there's apples in it, but there's other things too, other types of cells. And when we measure something in the blood or in the spinal fluid, that's sort of like this melting ice cream that's flowing onto the plate here and, somehow picking up some stuff. And so we think about these things a lot when we design experiments to validate. And is what's in the, the measurement in the blood or the spinal fluid reflective of the tissue? And as the disease progresses, maybe as the different types of cells change, maybe the proportion of ingredients changes in the apple pie. We also think a lot about, is an apple that fell off the tree the same as an apple on the tree? And are neurons that are left in somebody late in the disease, are they left there because maybe they have something that protected them? Maybe they're the ones who, somehow were protected. And so these are the sorts of things we talk about with researchers. And the thing of it is, I was thinking about like why it's what, so I spend my weeks talking to researchers all over and helping them get the samples they need. And, one of the things that's really amazing is that I'm able to do this without, like, I don't ask for anything. We don't ask for authorship, ownership, IP. We don't want anything back. We don't even want to be on the papers, right? And the only reason that we're able to do that is because of the support that we get from the CDC. And because of that, when we have these calls with researchers, because of this support, we're able to it's not about me getting a paper or a grant or something like that. It's what can we do to move the research forward? And, with the support from the CDC, we've relocated to Temple and we've been able to expand what we're doing. And Dr. Hyman Patterson had collected a bunch of autopsies that we've incorporated, and we're working on ways to be able to accept tissues from everywhere around the world with standard procedures and really doing things that we've never been able to do before. And again, when we get on Zooms, which we've been doing for a long time, way before the pandemic, we're able to just focus on what can we do to help. And I really am grateful that that's what I'm able to do, that I don't have to worry about how this affects me or my career or any of that kind of stuff. I can just think about how to move the research forward. So because of that, we say yes to everybody, right? And we make this clear up front. We don't say no to anybody requesting samples. However, we make sure that they're used responsibly. None of this would be possible without people living with the disease who then die and their families deciding to donate their tissues. And that's the ultimate donation. And so, we tell everyone that we talk to, we're going to make sure you get what you need, but we're going to do it in stages. We're going to send you pilot data. We're going to make your samples. We're going to make sure your experiments work, make sure that you get good results. We're going to use those initial results to help figure out what are the best samples after that. We make sure that this is not wasted, but we say yes to everyone. And I know there's probably people on the call who've had these conversations with me, and we are really, really meticulous about this and thinking about what's the point of the experiment. What are they trying to show? Is there a go, no-go decision in developing a drug? If so, what's the important measurement that's going to make that decision? Are they using the right thing to compare? And this is how I get to spend my time. And I just recently have realized that it's what you dream about. Like I've said before, when you're a kid, about what science is supposed to be. And I couldn't do it if it wasn't for the support that I get and for all of these resources that really have come from people living with the disease. So that mouse study that I showed you before, this is a study that, interestingly enough, originally was funded by the DoD, by the program, the ALSRP. And originally I was involved with this, now I'm actually part of this research team, but this is no longer that funded research. But originally I met this researcher, Dr. Zhu, because she had written a grant to NIH and they had said, you need to validate what you're doing in human tissues to make sure that it's actually happening in patients. And that was our role. So we contributed samples. And so what this is showing on the right here, this is the diaphragm, the muscle that moves air in and out of your lungs in a mouse. And these are ones from people who've passed away from either from ALS, from controls who didn't have ALS, from genetic ALS. This is a C9. And what I think you can see here is that in the diaphragm, the muscle cells in the mice, normal mice look like this. The ALS mice look like this. Again, normal mice, ALS mice, and the same sorts of stuff happened in the genetic and the sporadic people. And so this is how we can use these tissues to help validate these things and move them forward. In the past year, something else really exciting happened. So this gentleman, Dr. Garudo, who was at Binghamton University, had been curating this massive collection of autopsies that were collected by the NIH Center on Guam over many, many decades. So, there was this epidemic that started in the 40s and 50s on Guam of something called lytico-bodig. It was ALS. Some people got ALS. Some people got Parkinson's dementia complex. 10% of every death on the island of Guam amongst the Chamorro people in that time was due to ALS, and another 10% due to this Parkinson's dementia complex. And they collected all these autopsies, and they collected blood, and they collected spinal fluid. They collected blood serially over time as people went from being normal to then later having the disease. And they collected this information from their family members and detailed clinical information. And Dr. Garudo reached out to the CDC and said that, he wanted to know if they would be interested in this collection because, he was retiring and he wanted to make sure it was being maximally used by researchers. And the CDC reached out to us, and with funding from the ALS, the National ALS Registry, we've been able to incorporate these resources into our postmortem core, and we're going to, make them available to researchers around the world to study what caused this epidemic, how did genetics relate to environment, what is it that made these people more likely to get these diseases? How did Parkinson's relate to ALS? You know, and how did the changes in the tissue relate to changes in their blood? And so this is the kind of thing, it's just amazing to think about. Somebody called me up and said, hey, we've got this resource. Are you willing to help make it available? And of course, the answer is yes. And this is how, you know, this is the revolution. This is how things are changing. And nothing really exemplifies this more than what Everything ALS is doing. And, you know, this is one of these cartoons has to be me, right? I don't know. Where's the, anyway, I guess I'm the neurologist or one of them. So this idea that we have everybody working together and really in real time, and we meet every week with the data scientists and platform development to help move these projects forward. And, you know, the speech study for Everything ALS started before I gave that talk, before I was involved with Everything ALS. But after I joined was when we started doing this analysis. And, you know, I know there's people on this call who are involved in the speech study. The idea was, can we record people's speech on cell phones and all these different speaking tasks and somehow figure out a way using machine learning and artificial intelligence to tell when speech is changing better than our current methods, right? And our current methods are this ALS-FRS-R, and you basically have a scale from 0 to 4 of speech, from normal speech to loss of speech, and that's not granular enough to detect a change. And when the machine learning algorithms were seeing these changes, which they could detect, absolutely, we had to figure out what to compare them to and how to decide if they were meaningful. And we didn't have something to compare them to. And that picture down here, this was from Ernest Frankel's idea. He's like, you know, it's sort of the analogy would be if you invented the first thermometer and all you had to compare it to was like putting your hand on your forehead and saying, hey, do you have a fever? How do you know if your thermometer is measuring something that means something? And so we had to think about what's clinically meaningful about speech. And this was another analogy here, like, you know, the first airplane, if all you had to compare it to was dropping a rock off a cliff, well, you know, it's not granular enough. You got to know whether or not what you're doing is meaningful. And because of the way this Everything ALS community works, you know, we were able to mobilize this team of speech and language pathologists from ALS centers across the country. Jordan Green, who's an expert in speech and language pathology at MGH. And they sat down and they said, let's look at all the different measures of intelligibility and speech that have been used in any diseases. Let's figure out what might work here. You know, what could we use to possibly figure out what's a clinically meaningful change? And the point of this study was to get some data that we could use to then train these artificial intelligence machine learning algorithms. And it wasn't just about having those experts. You know, the team at Everything ALS, the Marcus and Julian, Esteban and Diego are the data analysis people. Swapno runs the platform team. You know, they were able to quickly produce a platform and a way to analyze this data very fast on the fly. And, you know, these are some examples. So what we ended up using, so we did, actually, I think that there's a slide, unless it fell out here. Hold on a minute. Let's see. Yeah. So, you know, look at this timeline. Look how fast this is. Look at the way that things are working in ALS right now. So we decided to try and do this in November of 2022. By January, we created an outline of the project, right, and what it was going to be and enlisted the SOPs. By June, we had completed 2 pilot studies looking at all different kinds of endpoints, different ways to present the data, to analyze the data, different platforms. What we arrived at was using something called listener effort. And basically what listener effort is a scale from 0 to 100 that those speech language pathologists were rating. And it's how hard is it to understand somebody, right? So how much effort does it take to understand somebody that's speaking? And if they're speaking normally, it doesn't take much effort. That's a 0. If you can't understand them, that's 100. What these plots are showing, these are 4 different examples of people in that data set. And the blue line is the ALS-FRS-R bulbar score, what we used to have. And the red is this effort scale. And so people who don't have involvement of speech are flat on both. But people who have involvement of speech, so this is looking over a year, right? And so this is somebody whose ALS-FRS-R score stayed stable and didn't start to decline until, you know, a third of the way through the year, and then it started declining. But look at this, the effort score picked up that decline much earlier, picked it up right away, and it was a steady change over time. This is one here where, you know, just to show it's not granular enough, somebody's bulbar score was stable, it went down a little bit, it was stable again. You know, our effort score gradually increased. This is one where somebody's effort score was, you know, self-reported, but didn't change at all. And yet this bulbar score, but this effort score did. It increased over time. And if I played some of these recordings for you, and I'm just going to play 2 of them for the matter of time, but.
Empty the trapped particles into your pan to recover the gold. Camphor in a day or 2.
And so I think you can hear that there's been a change in this person's speech between those 2 time points. It wasn't detected with our ALS-FRS-R score. It was clearly detected here. And that's the case for all of these. And so we have a manuscript that's about to be submitted, and it's analyzed all of this data. And this is just a little taste of some of it that's very fascinating. So using this listener effort score, 0 to 100, clearly we can tell between people with ALS and controls over time. If you look at when they enrolled in the trial, you can, people with bulbar onset ALS appeared to be going faster. But interestingly enough, if we only looked at people that had involvement of their speech when they enrolled in the trial, whether they were bulbar onset or limb onset, they actually progress at the same rate. And it's this steady rate. And so this is a granular scale that originally our idea was we were going to use this scale to train these machine learning algorithms. Well, guess what? It turns out that this outcome measure itself is useful and it's easy to do. And the SLPs could rate thousands of recordings in a very short period of time and get this really reliable data. And now we're going to look at whether this can work for international trials, whether SLPs in different languages can do the same thing. But also we could use that data in these machine learning algorithms. And this is just one example. This is showing how this artificial intelligence clustering could tell the difference between different people living with the disease, that some people it could predict that they were going to have no progression. Some people were going to progress slowly or a little faster or very fast. And so it really shows that this is the kind of measure that's going to help us measure the changes in people over time in much more granular. So, there's so many amazing things going on that I could talk about, and I'm not going to, but I wanted to give you guys some examples of where you can get information and where I get information. And this one, I'm going to call this person out. I don't know who this is, but this is a person living with disease that, hang on a second, has made an incredible resource. And if you're on this call, I'm a huge fan. Oh, hold on. Okay, it's loading. So why did it take so long to load? Because there's this massive thing, and I put that link in the slides. And if this person's, I don't know you, I don't think. If I do, I apologize. This is the person who's made this website. And they update this thing every week. And this is my personal go-to for information on therapies under development. Because if you go through these tabs up here, so see this tab called Meds, it lists like every development, and it has links to the companies, and it has links to the papers, and it has information on the publications. And honestly, I rely on this resource. I don't know who you are. I love you, and this is a wonderful thing that you're doing. This is how I keep on top and find information easily about all of these programs. Now, I don't endorse, there are some opinions here about what this person's doing themselves, and that's different. I was really, really flattered, by the way, when I went to snapshots and saw that's like my slide there. So whoever you are, I'm grateful for that. But they evaluate this by, different genes, by biology, by molecular functions. What are the genes? What are the the different other conditions that these things are used for? They analyze everything you could want. This is amazing. And again, this is somebody living with a disease who's doing something I would never have time to do, and I rely on them. And then beyond that, the ALS research program, if you go to the website for the ALS research program, there's a hyperlink to this file here of resources. And we keep track of all these things. So this is, where people can get information on all the different animal models and get them for their own labs, different animals, not just mice, all the biorepositories that are publicly available, right? All the different, so this is biofluids and cell lines. This is tissues, including our efforts, the Target ALS efforts, NIH, other places around the world, the Guam resources. This is data sets, data analysis tools, things like the ALS TDI data set, and of course the EverythingALS speech study data set, which is openly and broadly available. So this is like a one-stop shop that, if somebody wants resources, if somebody's applying for those grants and they want to know where they can get the things they need, antibodies related to the disease, clinical research tools and resources, the ROAD scale, which was developed by Dr. Fournier, platforms to search for clinical trials, right? Including the amazing ones developed again by people living with the disease, the ALS Trial Navigator, TDI, ALS Signal. These are things we all rely on. And so, and then I also wanted to just mention quickly, because more and more we're seeing people who have genetic mutations and their family members who are at risk and have them. And if you guys aren't familiar with End the Legacy, this is, should really be the go-to for resources to help people who are at risk, who have these genetic resources, these genetic mutations, to understand what that means, how they can get information on them, how they can get tested, how they can be involved in clinical trials and advocacy for people with these diseases. And I just wanted to mention too, so that ROAD scale, the new marker that was funded by the ALS research program, well, Everything ALS has now incorporated Dr. Fournier's, this new version, this thing called ALS Motor. This is a telemedicine, tool for tracking ALS progression. And they've made an app that lets people do this at home to use this score. And it just, when I sit back and think about in just the past couple of years, what I've gotten to see, this is what I meant by stories from the front lines, right? This is not my research. This is not anything that I'm doing. This is just breathtakingly inspiring that this new, we could get funded something to make a new outcome measure in ALS. And then not only does it get validated and, not only is it now something that can be used in trials, but Everything ALS is able to incorporate it and make it something that can be part of an app. And it's just remarkable to see this kind of thing. And it's not the way things were before. And by the way, if you want data on that, you can go to the Emory website. And so, this was a little bit all over the place, but I just really wanted to communicate to you guys how, this is not what things were like before. And therapies are starting to work, right? And we're figuring out who to use which drugs in. And we're sharing like never before. Now, this is my own personal wish list of what I would like to see now. And I want your help, because I've learned that if I say this in something like this, next thing I know, somebody's made it happen. So, so do your magic. One thing that I've talked about a few times recently, and this idea came from advocates, people living with the disease who first mentioned it to me. I really love it. The major barrier to collaboration between all of these data efforts and sample efforts and efforts that are, collecting diverse information on people is it's hard to share data, right? And it's hard to share data because it's all de-identified in different ways, and we have to protect people's identity. And, I would really love it if the CDC registry, it wasn't originally conceived in this data-heavy world that we live in now. Now we do. And if I think about what a modern day disease registry should be, it should be helping to foster the sharing of data across these efforts, not to be housing this data, but helping people share it. And so I personally would love it if the CDC registry was able to have funding to administer and really, a global, unique way that we can de-identify data that we all can use. Because right now, every researcher I know, every clinician, every basic science researcher, this is like such a hassle. And we all deal with all of our own, institutions, and they interpret everything differently. And there's no two ways that it's done the same. And, so that's one thing that I would love to see. Obviously, like everyone on this call, I can't wait to see what the Vision 2030 program does, because it embodies all these things we've just talked about from, Everything ALS. Me personally, so a couple things about trials. I don't want to see trials anymore that have a particular mechanism that's being targeted without a way to predict which people to give it to. I don't give my patients Qalsody, tofersen, the SOD1 therapy, if they don't have a mutation in SOD1. And I certainly wouldn't have enrolled them in a trial of a therapy for mutations in SOD1 without that mutation. We need to treat these other trials for non-genetic mechanisms the same way. We know that ALS is a lot of different diseases, and that every patient has, differing contributions from different abnormalities in their biology. And so if these trials are going to be successful, and if we're going to learn from them, we need to know that we are giving the right therapies to the right people. The ALS research program, the DoD mechanisms, we require mechanism-specific biomarker development to be part of those trial mechanisms. And this is why. And I want to see it. I really think we need to insist on this. Obviously, we want better ways to track disease progression. You know, my personal bias is that I think digital markers are going to be wonderful for that. And also they're going to help with improving equity, as I said, because we can do trials of people who aren't coming into clinic. We can follow their progression. We can follow at-risk gene carriers. You know, we need to insist that all of these clinical trials, as we've done with the mechanisms for the DoD, involve community collaboration. It's got to just be the standard way that it's done. And, you know, and these things are happening, they need to happen more. I wanted to mention too, so, you know, and this is, again, this is me speaking, not the DoD speaking, but so there are Department of Defense research programs in lots of different diseases. This is us, we're the ALS program, right? Some of them are smaller, some of them are bigger. The ones that have been really successful in getting therapies through from early science to effective therapies are these big ones. It's, you know, prostate cancer and breast cancer, and these ones that are really large budgets. And that's partly because they have mechanisms to fund later phase trials, which we just can't do. You know, trials are expensive. And, you know, I don't know what the future holds for the funding of this program, but my personal hope is that if there was increased funding, that it would let us fund these later stage trials. And you know, we know that that's important. And I wanted to use this example. So this is, you know, this Alzheimer's drug, donepezil, is often used as a case study of what's the difference between industry-funded late-phase trials and non-industry funded. And, you know, the point is that there is a bias to the way results are interpreted if a trial is funded by industry. And that's not, you know, that's not industry's fault, it's just a fact. And so I think that if we were able to play a bigger role, much like we've done, so by funding early phase trials, we've been able to insist that people involve, you know, people with lived experience. We've been able to insist that they have biomarkers because that's what you have to do to get our funding. If we had funding to fund later phase trials, we could similarly make sure that these trials are designed well and rigorous, because those would be the requirements of the mechanisms. You know, as far as further my wish list and what I'm most excited about, I mean, the stuff with the gene therapies, for example, what's going on at Columbia with the, you know, the personalized therapies for these rare genetic disorders is, I think, remarkable. I think figuring out which therapies to give to which people is really going to be the key. I mentioned this, that I want them to administer this unique identifier. This is actually the wrong slide here, but that's okay. And that's a duplicate. And, you know, and so this is what I'm hoping to see. And I wanted to close too. So, what am I personally planning to focus on besides, you know, continuing to hopefully grow these programs and work for you guys and do whatever I can to move research forward. And you know, I started off talking the last time about Dr. Griffin, who was one of my mentors who passed away. And so this is Fred Sachs. He was another one of my mentors. He recently passed away. He was my PhD mentor. He's the person who taught me how to do research. Taught me how to figure things out, taught me how to be a scientist. And when he died, a bunch of us who hadn't talked to each other in a long time, you know, all started sharing our stories on Facebook, on social media. And I wanted to share with you because I got to tell you, personally, what I plan to focus on right now is figuring out how all of these different ALS mechanisms, how all of these different broad genes, what makes it ALS? And I think we've got the tools to do it and the expertise to do it. And when I think about it, that's what I learned from him. And so that's my personal plan. So thank you.
Wow, Lyle, that was very powerful and a lot of information. And we've got a few questions. So Martha Beach and myself, we will ask the questions from the chat that we collected. And thank you for sharing the link on the chat here. And the first question is, you know, should we recommend that every person with an ALS diagnosis have a whole genome sequencing?
Well, so, I mean, this is part of, again, what's changed. Like I mean, I've only been in this ALS research for about a decade, right? And, you know, a decade ago, I couldn't get whole genome sequencing if I wanted to, and it was expensive, and it was academic because it didn't really change the way I treated somebody. And, you know, nowadays it's inexpensive and sponsored by these companies who are developing these therapies, and we're able to do it. And absolutely, I think that everyone should have, genetic therapy, genetic testing if they have ALS. Now, it's much more complicated if you're somebody who is a family member of someone who has ALS, and you want to know if you're at risk. And again, like, there's just too much information out there in the world right now. I will tell you, if you want to know how to handle that and what the issues are, go to End the Legacy's website. Okay, End the Legacy has unbelievable discussions and resources about what are the considerations for that. I did want to say one other thing too. So one of the reasons I was uncomfortable with giving this talk and into, I don't like being necessarily called an ALS expert, because what am I an expert in, right? I can explain the science, I can talk about how we do the science, but I'm not really an expert unless I can treat this disease, unless I can make you better. And that's really, and so I'm not comfortable with that. And the reason I wanted to bring that up is that a lot of times on these Zooms, I see, people asking, what do you think of this drug? What do you think of this drug? Is this a trial better than this one? And certainly I talk about this a lot with my own patients. And I'll just tell you, any ALS clinician or researcher who tells you, ooh, this is the trial you should do, this one's better, that's baloney, okay? We don't know what's going to work. If we did, we would have done a better job over the past 30, 40 years of translating these therapies. What we know is that we have to do a better job of figuring out which therapies to give to which people. If you want to be involved in clinical trials, and I greatly encourage you to do so, take whatever's offered to you, okay? Anyone that tells you one drug is better than another, there's just no way to know. I want every drug to work. I have no idea which ones are going to.
Wow. Thank you, Dr. Ostrow. That's incredible. I just appreciate how you're sharing your personal passion that's influenced your research. So we appreciate it. We have a question, very interesting, actually. In looking at the autopsies of people with rapidly progressing ALS symptoms versus slowly progressive ALS symptoms, have you noticed anyone that you know while you're studying these differences, noticing unique neuropathological finds?
Yeah, so that's a great question. And this is exactly the kind of things that dozens of labs are doing around the world, right? And so it's, whether it's, fast versus slow, or limb onset versus not limb onset, or a particular genetic type of ALS versus another type of ALS. And so we have to be very careful about how we do those comparisons, because think about this. So first of all, what makes ALS fast, right? ALS is a disease of progressive weakness, okay? What makes it a fatal disease is when it involves generally your breathing or your swallowing. If your disease starts in the breathing or the swallowing muscles, chances are you are going to, die from your disease sooner than somebody whose weakness maybe started in their toe. Not always, but maybe. When we're looking at autopsy tissues, by definition, they are end stage, right? It's somebody who succumbed to the disease. So whether it was fast or slow, they've succumbed to their disease. Sometimes when the people die, there's parts of their bodies that aren't involved. Some people die from ALS and maybe their arms aren't that weak, or one leg isn't that weak, right? And we have to worry also about, so again, at the end of the disease, is it possible that the pathological things that we see in cells that are left are actually the ones that are protected? If you have the apple tree, if a bunch of apples fall off the tree because the tree's sick, and at the end of this sickness, I'm only looking at the ones that are still on the tree, something kept them on the tree. They're not the ones on the ground. And so these are the sorts of things that we think about a lot with designing these experiments using the tissues. It's also why it's really important, like what everybody really needs is matched tissues collected after death to samples during life, like blood or spinal fluid or muscle. Muscle, by the way, has nerve endings in it. You can get, motor neurons out of it too. We need to know how what we find in the tissues relates to what we see with people as they progress over time. One reason I'm super excited about this collection that we got from Guam is that it includes the blood collected serially over time while people were alive, and the autopsy tissues after they died, and their family members, and some people who became symptomatic, who started off as controls. And so those are the ways that we're going to be able to answer those questions. Now, interestingly enough, when we talk about fast versus slow, so one of the slides I pointed out for a moment there with the EverythingALS speech study, everybody's always thought if you have bulbar onset ALS that you have fast ALS, right? What we found is it doesn't matter if the onset is with speaking or with your limbs. if at the time that you enrolled in that study, you had some involvement of your speech, you progressed at the same rate. So these sorts of digital measures are going to help us figure that out too.
Excellent. Yes, thank you. I found that fascinating. And I was trying to read some of the notes on that slide so from Guam. And so I thought that was fascinating, that the qualitative piece that's being added. So thank you.
Perfect. So the next question was, where do we get the information? Because we want a single source of truth. That would be a great vision. Is there work being done to funnel some of these go-to places? Lyle, I'm going to take this question as you come here, exactly what Dr. Ostrow said. This is why we have this expert talk series. You can come here, and because it's very hard to figure out the single source of truth. And with that, I'm going to just have...
hang on. Can I add something to that into that I think is really important? Okay. So look, nobody knows what the actual total truth is, but you do want to have responsible sources. But also, like as an example of how these different programs are interacting and helping each other, if you do the Clinical Research Learning Institutes, you learn how to read a research paper, right? That's part of what we teach you in that. You learn how to read a grant proposal so that you can be a better reviewer for the DoD ALS research program, okay? In this data-heavy world that we live in, there's a lot of people out there who want to take advantage of desperate people, right? And the internet is filled with bad resources as well. And things like ALS Untangled are wonderful resources to find on those drugs. Nowadays with preprints, right? So nowadays you guys find out about research even before it's been peer reviewed by scientists. I was asked to be on a panel during COVID of the impact of preprints on COVID science, on the how rapidly the science developed for how to treat COVID. I'm not a COVID doctor, but they wanted me on there as an ALS researcher to talk about how it impacts just research in general. And the fact is it speeds it up because people are sharing the results quickly with other researchers can use them. The problem is we have to learn how to be responsible consumers of that information. And the analogy that I used in that Zoom was it's like when the first cars were invented, right? And so when there were horse-drawn buggies and everybody was using horses and then cars came along, cars were much faster. You could get from point A to point B super fast. It's literally a way to get somewhere faster. But those early cars, they blew up and they crashed. And, they had to figure out not only how to make them safe, but how to train people to be drivers. And I have a 20-year-old kid, and oh my God, did he need driver's ed? And so we have to do the equivalent of driver's ed for understanding how to consume information in this data-heavy world we live in. And Everything ALS is doing a wonderful job of curating this. So are many other resources out there. I have if anybody wants, I have a list that I give to people. But the fact of the matter is, it's not about who's going to tell me the truth. It's about learning how to consume information responsibly yourself, and then training others in the community to do so.
Yes. And Lyle, if you don't mind, I'm going to call on my colleague Sylvia to just talk about the research that we are just launching, which is, one of the things that Dr. Ostrow actually mentioned about end the legacy for people who are at genetic risk. And this is the study that we are doing. So Sylvia.
Hi, everyone. It's so good to see all of you. I actually see a couple of familiar faces. I'm Sylvia. I run the research portion of Everything ALS, and I just wanted to share my screen and talk about a new study that we're rolling out. We're actually, if you have followed us for a while, you probably have heard about our Radcliffe and our speech study. We are actually now recruiting for our asymptomatic study. The asymptomatic study, we are recruiting gene carriers. And so if you or if you know anybody that has a history of familial ALS that is currently asymptomatic and over the age of 18, we got IRB approved. And so we were first in the the design phase, and now we're IRB approved and ready for recruiting. Couple of things about our study. It's 2 years, and it's done at the comfort of your house. We actually have a speech portion of the study and also a motor function portion of the study. And so it's done at the comfort of your house with a proctor or a coordinator like myself. We schedule a call once every 3 months, and the call is about an hour long. And all that data that we collect when you do these sessions with us for period of 2 years on this study, you actually are able to see it on your participant dashboard, on our secure participant dashboard anytime. And so the study is really for, people that are asymptomatic and gene carriers who would like to contribute and just, be able to see their own data in one place. And so we have that study rolling out. A couple of you I see that are familiar faces have also been a part of our speech study. Our speech study, we enroll for participants that are diagnosed with ALS or probable diagnosis with ALS, but also for participants that, people in our community that, don't have ALS and are family members or caregivers or daughters and sons that just want to, help and know more. This is a great study if you're someone that, doesn't have ALS, but would still like to contribute and research. It's called the speech study. And basically we onboard you onto this study, and it's just speech and video tasks. It'll record your speech through speech task sessions. You record for about a year at any time at the comfort of your house. The first session usually takes about 20 minutes, and then you could just open your desktop, log in through our app or log in through our EverythingALS, sign up, and just be able to onboard it. You don't have to onboard yourself. We or myself, I usually do most of the onboarding, will onboard you onto the study, and then you could do the speech tasks once a month for about a year, and you can do it even frequently if you would like, maybe once a week. So those two are our studies that we're really recruiting for, and we would love to have you, or if you know anybody that would, like to join, because our research and the data that we collect is all because of you guys. So I really appreciate it, and thank you for giving me the time to speak about it.
Thank you. So switching back to Dr. Ostrow here, I think I'm going to ask another question, then let Martha do it. One of the questions that came up, Lyle, as you were talking, was how and when do we eliminate placebo in the trials? 6 months on the sugar pill, for being on the placebo for people with ALS is a lifetime. So in your mind, when can we be in a place where we can actually, eliminate placebo?
So a couple things. The real answer to that question is I hope that someday we have drugs that are so obvious that they work that we don't need any of this stuff, right? And I really think that that's possible. Placebos are really important. They just are. And, the standard of care in ALS, I mean, if you are followed by a multidisciplinary clinic, if you are in a trial, I mean, these things change the trajectory of your disease. Because, for example, over the past 35 years, if you were in an ALS trial, there are 3 possibilities, right? You could stay the same, get better, or get worse. And believe it or not, there were several clinical trials which sped up the disease. And when we went back, I mean, right? So things that worked in a mouse, and then we brought them to people, they sped up ALS. And when we went back and looked, what we realized was that those were drugs that were causing weight loss. And that was the beginning of this idea that we got that, losing weight speeds up ALS and maintaining your weight slows it down. And why a lot of us now advocate for really staying on top of the nutrition, or maybe early PEG tubes, whatever it might be. And if you're in a clinical trial and you're being seen more frequently, it impacts that kind of thing, right? And so I personally think placebos are important, but the thing is with, for example, the Healy, the platform, right? That's a way we have figured out how to test lots of drugs at once that can share a placebo. And, that's the way that we're going to be able to do research faster and do it with less people on placebo. But my personal opinion is we're not going to be able to do away with them until we have therapies that legit work, where I don't need any of this stuff, where, I can clearly see whether something's working or not.
Thank you.
Thank you, Dr. Ostrow. We had someone in the chat, you were talking about the pipeline, and that the pipeline process, they said it could work really well at times if you have an understanding of the underlying mechanism of a disease. So with ALS, in terms of the mechanism, without this understanding, how are we going to make progress? What's your thoughts about that in terms of where we are with understanding the mechanism of ALS?
Yeah. So first of all, I want to be very clear. Like, I'm not saying blow up the pipeline as in stop doing this, the ecosystem the way we have it designed. I'm saying I'm offended by the word pipeline. And I have trained myself over the past 2 years since I last spoke to this group that I shouldn't call people living with ALS patients. And I agree with that. And I've worked very hard to burn something out of my brain that has been there forever, right? And, you know, and what I'm saying is I'm offended by the word pipeline because it doesn't represent what we're doing right now. And so that's when I'm saying, you know, the pipeline, look, we still need to follow this pathway from basic science discoveries through to clinical trials. But the point is, I don't think that's represented well by the word. And so somebody has to give me a better word. Now, as far as how we can improve it and do a better job, it's happening, okay? And the way it's happening is as follows. You know, first of all, the purse strings from the ALS research program, by refining our mechanisms, even for the earliest science, we now require people who are doing basic mechanistic science on a lab bench, like some new biology, even at that earliest level, they have to be thinking about what the biomarker is going to be in their eventual clinical trial. That has to be part of what they're proposing to us. This is the way that we're going to improve therapy. We know that ALS is a bunch of different diseases, and we know that, you know, that they combine differently in different people. And when we analyze genetics or tissue samples or RNA or, you know, proteins, that we see these clusters, that some people have different abnormalities of biology that are dominant. And so right now, the biggest improvement in my mind to the pipeline is that we're paying more attention to that so that we don't do something in one mouse and say, oh, let's give it to everybody with ALS and see what happens and have another failed trial and not know. And, you know, afterwards say, well, maybe it worked in some people, but we just don't know who they are, right? And so that's the biggest change, I think, right now to the way that we are doing therapy development. The other one is sharing data, legitimately sharing, legitimately collaborating. And as I said, though, my personal feeling is the thing that could dramatically tomorrow improve that is if we had the CDC administering a global unique identifier that was truly global. And by the way, doing so would also improve the catchment of the CDC registry. Because if you had to go to that website, you know, or your doctor had to go there to get your assigned de-identified, you know, GUID, then instantly it's going to improve the number of people who are signing up on the registry. And it's going to mean that we're capturing all the trials and all of the studies and being able to link this stuff. So that's one thing I think will improve things a lot.
Thank you so much. Well said. Thank you.
Thank you. And I think we have the last question here, which is, what therapies do you recommend right now for people with ALS? And the second part to that, yeah, that's the first part. And the other one was, do stem cells really show any promise?
Okay, so the first part of that question, my job as a clinician is to inform my patients, right? And so I need to be able to explain to them for an approved therapy, what's the data, what's the risks, what's the benefits, and then you decide what you should take. What I will say is that I want every patient that I personally see on Rizol. Rizol, it's not a cure, but it works. It slows the disease down. It slows it down better if it's given early in the course of the disease. Every trial that's ever been done, right, because there's placebo and there's drug and some portion of the people are on Rizol or not, has shown that it slows the disease down, okay? You know, approved therapies for this disease, they have different mechanisms. Radicava works differently than Rizol, okay? What we don't know, but we're starting to get a signal, is perhaps like 1 + 1 = 4. Perhaps being on different therapies targeting different mechanisms will be beneficial. As far as, you know, other therapies that are in development, again, I don't know what's going to work. I truly believe that at some point in my career, I will probably use stem cells engineered to make some factor, whether it's a growth factor or something like that, in treating ALS. Okay, but I don't know when that will be. And therapies that involve infusions or you know, more labor and cost to deliver, we have to be careful. I want everybody to have access to them, but we got to know who to give it to. We got to know who's going to respond and who's not, because our clinics simply don't have the bandwidth otherwise, right? I want to be able to take care of every patient as best I can. And the only way we can do that is if we are rigorously figuring out which people living with the disease respond to which therapy, because we know it's not everybody for any of these therapies.
Thank you. I think one last question. I know it's very dear to you. Is it true that ALS is not a mandatory reportable disease like MS and Parkinson's to the CDC?
That is true. In some countries it is reportable, and in some countries it's not. And you got to be, there's some other issues in this too that, I mean, it's a long discussion. I'm happy to have over a virtual beer sometime, but ALS is a clinical diagnosis, right? So it's kind of hard to figure out how you would make it mandatory. I mean, I know in some countries they do, and if the clinician says that it's probable ALS, fine. But aside from the genetic types of ALS, the only way to definitively tell that someone has it is an autopsy. And we're pretty good at diagnosing it. We're not so good at getting people to an ALS neurologist fast enough to get the diagnosis done. But, I think that there are barriers to making a disease reportable that doesn't have a simple way to diagnose it across the board, is what I would say.
Thank you. Thank you so much, Lyle, and we really appreciate your time here. And gosh, a lot of information and a lot of good, fantastic, feedback and presentation. So thank you so much for taking the time, out of your
and I mean, just last, I mean, I know a lot of you I know, and I mean it, like you are my personal connection to ALS. I don't have a family member that had this disease or a friend growing up. And I am, and I say this a lot, I'm ticked off that I'm not doing a better job, that we, the scientific community, haven't figured this out yet. I got to tell you, a few years ago, I think that the mood was not good. I'm more confident than ever that we're going to beat this thing. And I cannot believe, I wasn't kidding. Like, if I don't check my email for a day or social media or whatever it might be, I think I'm going to miss something exciting and important. And it's just remarkable. And so thank you all. And if there's ever anything that I can do, please let me know if I can, anything at all.
And I know that, Michael, you have a question, so we'll ask you to unmute and ask your questions.
Dr. Ostrow, I'd love your opinion on Relyvrio. Are you disappointed, or did you expect something like this to possibly occur, to pull it from the market?
So I'll just get back to the same thing I say, which is that I suspect that Relyvrio works for some people, and I wish I knew who. And, we think we know the mechanisms of that drug. And my personal feeling is that every drug in development in ALS should have a mechanism-specific biomarker that we can tell, is it impacting, first of all, so for example, what I mean by that, if you have a drug that's targeting, mitochondrial trafficking, you'd like to know which people living with disease have a problem with that, right? And you'd like to be able to know if, when you give the drug, did you affect that thing? And without mechanism-specific ways to measure an effect, without mechanism-specific ways to figure out who's going to benefit, we're not going to know. And there is no way to know. Some of the trials right now, so for example, the HERV-K, the endogenous retrovirus study, we're able to measure things in the blood that suggest who might be more likely to respond. That's true for some of the inflammation-related therapies as well. We're getting better at this. We need to do even better. Nobody's going to enroll somebody in a gene therapy trial that doesn't have the gene mutation. We have to stop thinking that, we're going to have a successful trial if we're not doing the same thing for these other drugs.
Thank you.
Thank you. And with that, I think we'll open up an open forum, and Lyle will return the rest of the evening back to you. And this is the place where, you can stay as long as you want. We actually have everybody have conversation, and we'll actually open up the open forum. And McFinn,
Thank you,
I'm
everybody.
out, by the way, unless you kick me out, there's nowhere I'd rather be. So, but I joined anyway.
Yeah, thank you.
It's an extended family, folks, and even the doctor knows that he's part. When you have ALS, there's somebody out there who wants the brass ring, and they're going to find the answer. And it just might be Dr. Lyle.
Yeah.
We have no idea. So folks, don't ever give up hope. Come and join your extended family as often as you can, because to your living room comes this information that is priceless, because that's how important it is to each of us to hear what's going on. How can maybe we be a part of the research? That's a big part. But one of the biggest parts is when we put a smile on our face because we know somebody's caring about us. And tonight we heard there's a lot of care going on. All right. So folks, it's that time of day. It says that it's 5:32. So let's just say good night. Find somebody that you see here that you'd like to say good night. And say good night to them. This is your
night.
family.
Good job, again.
Good night.
Where's our hearts? Hearts.
That's right.
Yeah.
Good night. Thank you, Lyle.
Good night.
We love hearing your voices. Oh, Patty Hancock, I love you. Thank you.
Good night. Good night. Thank you. Good night.
Good night, everybody.
And Jim, your hero. All right. Good night, Jacqueline. Thank you for coming. It means a lot to us. Keith and Rita, thank you for joining us tonight.
It was a pleasure. Thank you.
Thank you. And Jubilant. Jubilant, you make my day every time I say your name. And Chuck Shaw, I see an angel right next to you with a beautiful smile. You're a lucky man.
I sure am. Thank you.
Yes, sir. It shows. All right. Sean, good night.
Good job, guys.
It's a beautiful night.
See you next week.
That's right. Lynn, thanks for all those comments tonight. John Michael, you're my hero. Thank you. Keep it up. That's right. Eric's job is to make people happy. And Kathleen, we'll see you again, hopefully next week. Okay, Vivian, keep up the good work. We'll see you shortly. Good night, everybody.
Night.
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 Kisses and love. Bye-bye.